Okay, let's do it. Just as a reminder, we have the Q&A open. So if you have any questions during the talk, I'll have that open on my screen and can try and address those as they come up. My name's Benji. I'm from the Allen Institute for Cell Science, and this workshop is going to be on our tool called CytoDL, and it's for versatile deep learning analysis of 2D and 3D images. So let's see. Getting my slides going. Yeah, so I'm coming to you live from the Allen Institute for Cell Science in Seattle, Washington. We're a nonprofit biology research institute, and we form a highly multidisciplinary team of about 80 people. Our core values are team science, open science, and community empowerment, and these values inform the tools and data that we release. So over the past decade or so of our existence, we've released tools in areas ranging from HIPSC, gene editing, imaging, computational analysis, infrastructure and modeling, all the way through data visualization and sharing of data. I'd like to encourage you to explore some of our other cell analysis toolkits as well. We've got gene editing tools, microscopy tools, analysis tools, and you can see all those on the right, and you can find all this and more at our website, which is allencell.org or alleninstitute.org. So today, I just want to briefly introduce the data that we're going to be working with. So I'm going to do like some brief introduction slides, and then we're going to have a collab notebook where we can get into some model training and looking at some data with CytoDL. But the data that we're going to be using is from one of our papers from a few years ago where we did this drug perturbation pilot study. So we have a bunch of different cell lines that we've tagged with endogenous GFP for a lot of the major organelles in the cell. And basically did a matrix where we analyzed each of these cell lines with a bunch of different drugs that are known antagonists or agonists to different pathways at different concentrations. And then we performed high throughput 3D high resolution imaging of each of these combinations of drugs and organelles. And then it gives us this nice matrix of drug by organelle that we can analyze these images and try and figure out what's going on in these cells. Let's look at more closely what one of these examples in this matrix actually looks like. So here we're showing some examples of our microtubule cell lines, our myosin cell lines, actin and Golgi, and then our vehicle, and then our two of the example drugs are paclitaxel and staurosporine. So for each of these cells, we're tagging the nucleus and the DNA. We're getting the cell membrane, and we're also tagging each of these individual organelles. Here's the paper that this data set comes from on the top right, if you'd like to check that out. But the goal was to identify signature profiles of cellular organization for a variety of well-known agonists and antagonists in kind of a novel way through this deep generative model of 3D single-cell organization. And the results was that we largely confirmed a lot of the known effects of what structures these different drugs target. So today we're going to be revisiting this data set in even a different way from how this paper approached it. So the data that we're actually going to be using for training is we had about 40,000 3D spinning disk confocal microscopy images. And each of these FOVs contains the plasma membrane, which is dyed with a cell mass deep red dye. We're labeling the nucleus with nuke blue. Then we have one of these 24 subcellular structures. And we also have cell instance segmentations that were generated for this publication, and we're going to use those for training an instance segmentation model. And then from this, in addition to this data set, we also have a single cell data set, which uses these cell instance segmentations to generate about 27,000 single cell images, each with one of these 24 subcellular structures tagged. And these were not treated with drugs. So this is the data we're going to be training with. First, we're going to train an instance segmentation model, and then we're going to train a contrastive learning model to do some unsupervised feature extraction and visualize what's going on with all these drug perturbations. So that's the data set we're going to apply it on, is this drug perturb data set. So we have 2,000 single cells with one of only 5 subcellular structures, and we're going to look at that with 4 drug treatments. So the goal of this workshop is to show you how CytoDL has been useful for us internally, and hopefully get you thinking about how you might be able to use this tool as well. As I mentioned, we're going to first train an instant segmentation model to generate a single cell data set. And then we're going to extract unsupervised features from these single cell data sets for detecting and interpreting the effects of the drug perturbations on cellular organization. But now you're probably wondering, like, what actually is CytoDL? It's a Python package on GitHub and PyPy that we've created to make deep learning microscopy image analysis projects more modular, more maintainable, and more reproducible. And what this comes down to is identifying the common structural components of a deep learning project. So this is your data loaders, your models, your callback code for executing arbitrary code within your training loop, that sort of thing. The second part is providing new components when they don't already exist. So there's a lot of microscopy specific, data augmentations or types of data loaders or image readers that aren't super common in the general Python community, but are really useful for us that do bioimage analysis. So we provide these new components when we need to, but CytoDL aims to make the reuse of already existing open source components a priority whenever possible. And then we also provide what we call recipes for training for kind of new types of combining these components. So this could be like the training loop for a GAN or a training loop for an instant segmentation model, that sort of thing. Okay, so let's get into the actual Colab notebook. Here's the URL. This should take you directly to the Colab notebook. The last letter is a capital O and not a 0. So I'll give everyone a few seconds to open up this URL, and then I'll switch over it to myself. Okay. So okay, that's not great. Could someone post in the Q&A if they're able to access this URL? Okay, now it seems to be working for me. All right, so before we get started diving into this notebook, we're just going to change the runtime to the T4 GPU. And this is just because we're going to be training a few small example models, and it's a lot faster to do that on the GPU than on the CPU. Okay, and then there are a few commands that are going to take a few minutes to run, and we just need these to set up our data set. So this first one is going to clone the repo that this notebook lives in and install some dependencies. If you trust me, you can hit run anyway. The next thing we're going to do is, since, we don't have time to train up some really good models during this workshop, so we pre-trained some models for the instant segmentation and the contrastive models. So this next cell is going to download those and make them available to you locally. And then we're also going to download some of the single cell data set that we've pre-computed for visualization purposes later on. So that's what that second cell does. And then this data set is available on Quilt, which is kind of a wrapper around S3 that you can access programmatically. It's commonly used for releasing data sets, and this particular data set is released on Quilt. So we're going to download a few examples from the raw drug perturbation data set as well using Quilt. And that's what this third cell is going to do. So while those are running, while we're downloading all of the models and everything that we need, I'm going to do kind of a more in-depth overview of CytoDL before we get into looking at the details. Is this working for everyone? I hope that you can use the Q&A if you have any questions or if something is not quite working for you. Okay, so as I said, one of the goals of CytoDL is to identify the common structural components of deep learning projects. And this graphic kind of lays out what we mean by that. So we've figured out that kind of the core components of deep learning projects are the aspects of the data. So this could be the type of data that you want to load, and some microscopy image specific augmentation tools. Then you have your model, which is not just the neural network architecture, although that's very important, but also how the data flows through the network. So if you can imagine you're doing kind of a supervised learning task, like instance segmentation or semantic segmentation or super resolution, something like that, where you know the ground truth that you want to output from your neural network, the data flow through that model is very different, and how you calculate the loss is very different than something like a contrastive model. Another important component is the callbacks. So this could be something like model saving or evaluating the gradients, looking at the gradients and visualizing them. This is another key component of deep learning projects. And then the trainer, which handles the optimization. There are a lot of hyperparameters there that you might want to keep track of. And finally, logging. So each of these components is kind of a unique part of a deep learning experiment. And what we do in CytoDL is we have a single configuration file for each of these components. So you have one configuration for your data, one for your model, and so on. And then what a deep learning experiment means to us is a unique combination of each of these configuration files that fully specifies the code that was run and the model that was trained by your deep learning experiment. So these configuration files are hierarchical in that we have a single configuration for the experiment that compiles and resolves all of these sub-configuration files into a single source of truth that you can use to fully reproduce your deep learning experiment. So this is a nice idea, but how do we actually do it? So we rely heavily on two different libraries for doing this modular configurability. The first one is Hydra, which is a library from Facebook that lets you kind of operate with Python objects from within a configuration file. And this is what really makes this framework really modular. You can easily switch between different models, data sets, or training configurations. And using these configuration files, we provide project templates that should run out of the box to quickly train 2D and 3D models for in silico labeling. Okay, sorry to interrupt myself, but when it says restart session, you don't actually have to do that. You can just hit cancel, and that'll be fine. Okay, so we provide essentially ready-to-go projects for 2D and 3D image models for in silico labeling, so predicting a fluorescence channel from a bright field channel, semantic and instance segmentation, classification, contrastive learning, and masked image modeling. And we do this through the configurability of our config files. So if we look at what Hydra actually does, it's a tool that we can use from within our configuration files, and it relies on this _target_ syntax that you're going to see a lot in the configuration files as we get into them in a little bit. So basically what this does is it lets us operate on Python objects and specify them from within the configuration file. So if you have this _target_ numpy.array in your configuration file, basically what this means is when this configuration file is passed into CytoDL, it's going to instantiate this numpy.array object. Everything that's under the target syntax is going to be a keyword argument to the function or the class that you've defined in your target. So here, this whole configuration, this little YAML file, is exactly equivalent to the Python code calling numpy.array on a list of 1, 2, 3. So we're going to use this ability to work with Python objects a lot in our configuration files. The second tool that lets us make these really modular configuration files is OmegaConf. And in particular, we use this interpolation syntax, which is $ and then curly brackets. And this allows for us to interpolate variables in the configuration files. And this lets us kind of have consistency across configuration files. So for example, if you have a variable in one configuration file called path, and it has the path to your data, and then you need to access that in two different parts of your configuration file, instead of copy and pasting that path over and over again, which makes it easy to introduce bugs, you can use $ and then path in curly brackets to refer to the value of path. You can also use this to enforce consistency between different configuration files. So if a variable is in your data configuration file, you can access it using data.variable with this interpolation syntax, or model.variable, or whatever configuration file it's actually in. Okay, so I should also mention at this point that we primarily use CytoDL by cloning it from GitHub and then using it through the command line. But for the purposes of making this notebook easy to operate with, we've also created a simple API that's very much in beta mode, but it lets us operate in much the same way. So how we're going to use this is we're running this first cell where we import this CytoDL model object from the CytoDL API. We can instantiate it, and then we can load its configuration file from just a YAML file that we downloaded earlier in one of those first 3 cells. You can also load a configuration file from either a dictionary or use one of the provided templates for 2D or 3D models across those multiple tasks that I mentioned earlier. When you're loading the configuration too, you can also change some of the configuration parameters, and we'll get into that in a second. Okay, so the model that we've loaded is the training configuration for an instance segmentation model. So let's take a look at what this actually means. So with a data configuration files, the 2 things that we want to specify are how do we load our training data and how do we load our validation data. So in the data configuration file, you'll see that there are 2 keys, train data loaders and val data loaders. So these are essential for specifying how we're actually going to be loading our training data and our validation data. And then underneath that train data loader key, we'll see this target syntax from Hydra. So what this means is we're basically going to import this make array data loader function from cytodl.datamodules.array. So if you remember back to the Hydra syntax, that means we have kind of this Python function available, and then everything below it is going to be a keyword argument to that function. So there are some kind of basic data loader arguments that we'll pass in, like the number of workers that we want to use or the batch size. And then we also are specifying the transforms that we want to apply to each object that's being passed through our data loader. So one thing that's key to understand is this make array data loader expects our data to be in the form of each object in our data set should be a dictionary. In this case, our dictionary is going to have 2 keys. The first one is going to be raw, which is going to refer to our raw microscopy image. And the second is going to be seg, which is going to refer to our instance segmentation ground truth that we're going to be using for training our neural network. So this make array data loader is going to take in a list of these dictionaries, and it's going to perform all of these transforms that we're listing below here in order on that data. So notice that with this target syntax, we can mix and match where we're getting our functions from. So here we're calling a function from CytoDL. But then if we look at our first transform, this is actually from the MONI library, which is a super helpful library for bioimage analysis with deep learning. And we base kind of all of our transforms off of this MONI library. So this first transform that we're going to do is we're going to cast our NumPy arrays that we're passing in into tensors. So what's essential about this syntax is this keys argument. So this is specifying which keys in the dictionary that we pass in we want to apply this transform to. So you can imagine there are some transforms that we only want to apply to our raw image and some that we only want to apply to our segmentation image. In this case, we want to cast both the raw and the segmentation images to tensors and convert them to 32-bit floats. So after we've converted those images to floats, we want to do some image normalization. So maybe there's like a hot pixel in our raw image or just some crazy extreme values that we want to clip out because they're not super relevant for doing this instance segmentation. This CytoDL transform is going to clip out the top 0.1% and the bottom 0.1% of these intensity values. And then again, we're mixing and matching between these CytoDL transforms and these transforms from an external library because we don't want to reinvent the wheel. But this MONI transform will just subtract the mean and divide by the standard deviation of our raw image. So yeah, it's important to remember, like, this is not a transform that we would want to apply to our instance segmentation image because that's just a bunch of labeled objects. So we can specify with the keys argument which image we want to apply this to. And yeah, it's often helpful to have the documentation for Moni or whatever library you want to be using kind of up at the same time when you're creating these configuration files, just so you know which keyword arguments you want to pass under the target keyword. All right, so now we've normalized our raw image. The next thing we want to do is we want to take our instance segmentation, where we have each cell labeled with its own unique ID, and we want to turn this into something that we can actually optimize to produce using our neural network. So this kind of object labeled setup for an image is not ideal for producing a real instance segmentation during training, because these integer labels are not actually semantically meaningful. They're kind of random. So we want to turn this instance segmentation into something that we can actually train a model with. I'll get into what this actually looks like later, but this is just an instance seg preprocessing transform that we have in CytoDL, and I'll explain what each of the components of that are later. Okay, so the next thing that we want to do is our input so far has been full FOVs. And because we have limited GPU memory, we don't actually want to train our network using these full FOVs. They might be different sizes, and that would break things, or they might just be too large for our GPU. So we want to actually crop small cubes or squares, if you're in 2D, out of this large FOV and train the model just on those randomly selected little cubes. So this is something we want to apply to both our raw image and our segmentation image. We want to make sure that they're always the same size, so we set random size to false. And then we're just sampling one random crop in this case, but you could set that to be more if you have a bigger GPU or something like that. So this is the first time we're seeing the OmegaConf interpolation syntax for the ROI size. So this says in our data config, we should expect this aux key, and under the aux key, we should have a variable called patch shape. And this will tell us how big we want each of our randomly selected ROIs to be. So if we scroll up, we'll see we're in the data config already, and we look under this aux key, and then what do you know? We've got a variable called patch shape. In this case, it's just 16 by 16 by 16. So for the purposes of this tutorial, we're just going to take these tiny little crops from our image. If you're actually training a model that you want to be good, you would probably not make this exact choice. So basically what this syntax is saying is that our ROI size is going to be 16 by 16 by 16. Okay, so now we have randomly selected crops from our raw image and our preprocessed segmentation image. And then we might want to do some augmentation of those images. This could be something like rotation. In this case, we're just going to do some random flips across the Y and the X axes. So overall, basically what we've done is we've taken this makeArray data loader class, which takes in a list of dictionaries. The dictionary has two keys, raw and segmentation, and the values for those are some NumPy arrays. We've specified the list of transforms that we want to apply to each of those keys. This is very flexible. You could kind of make this whatever you want. And then we're ending up with these 16 by 16 by 16 randomly selected cubes from our raw and our segmentation images. I'm not going to go through the val data loaders because it's basically the same exact idea. We just don't do the random augmentation at the end, so I'm going to skip that for now. Is everyone feeling good about the data configuration file? I'll assume that's a yes until a question pops up in the Q&A. So that's loading our data, our data preprocessing pipeline. Next, I'd like to move on to our model configuration file. So the model configuration file determines how, once we have this data loaded, we want to kind of pass it through our network and optimize our network in a very specific way. And that depends on the task that we want to do. So we're going to use a class called multitask im2im for our model in this case, which creates a single backbone network that'll take in our raw images and create a shared feature representation, which can then be passed to any number of task-specific heads. So say you have a 2-channel image, say of the cell in the nucleus, and you want to get both a cell segmentation and a nuclear segmentation. You would have this backbone network, which takes in your 2-channel input, that outputs this shared feature representation, and then you could have a task head for your cell segmentation, task head for your nuclear segmentation, and you can keep stacking these task heads for as many things as you want your model to be able to do. So each task head will produce a different output that will be optimized based on a task-specific loss, and the ground truth being used for each task head will be specified by the name of the task head. So in this case, our task head is going to be named seg, and that just tells this model that it should be using the seg key that we generated earlier from our dictionary as the ground truth for optimizing this particular task head. Each task head also has specific post-processing arguments. So the post-processing for a semantic segmentation might be like running sigmoid and doing a thresholding. Whereas if you're doing like fluorescence intensity prediction, you might want like hard tanch or something as your output. And for your instant segmentation, you might want a whole different post-processing setup. So this kind of flexible design lets us have a bunch of different task heads, each with its own loss function, ground truth, and post-processing. All right, so that's the general understanding of how this multitask M2M model actually works. Let's look at it in the configuration file. So the first thing you're going to see is we're using this target syntax again to instantiate this multitask M2M class. As a reminder, everything under here is going to be keyword arguments to this class. So we can pass in how often we want to save example images. Here we're going to set that to 1. So after every epoch, we'll save out an example of how the model's doing. This is really useful for checking your model performance. We also want to pass in an X key. So this is again going to be connected to our data config. So this is going to tell our model to pass the raw key into our neural network instead of the seg key. And then we're going to specify this backbone network. So here again, we're going to rely heavily on open source stuff like MONAI. We do have some custom networks implemented in CytoDL, but when MONAI has already done all this nice work to implement these state-of-the-art networks, why reinvent the wheel? So our target is going to be this dynamic UNet from MONAI, which is based on the NN UNet framework. And then everything under this is going to be keyword arguments. So based on what size of model you want to use, the spatial dimensions of your data, all of that, you can essentially craft this convolutional UNet into whatever you need for your backbone network. One thing that's important is, since we're not actually predicting our output from this dynamic UNet, we're just producing a shared feature representation. Our out channels is going to be a parameter that we want to keep consistent. So the number of out channels for our shared backbone should be the same as the input channels for each of our task heads. So that's why we use this OmegaConf interpolation syntax that just keeps everything consistent across our config. And then we can specify the size of our network, the strides, the kernel size for our convolution, and the number of filters in each layer. So that's our backbone network. Now let's specify each of the task heads. So again, the name of the task head is going to specify which of the potentially many ground truths from our data config is going to be used to optimize this specific task head. So here it's seg, and we're going to use this resblocks head from CytoDL to transform those shared backbone features into an instant segmentation specific output. But what this resblocks head does basically is a bunch of convolution with some residual blocks between them. It takes in the same number of channels that our shared backbone output, and it's going to output a 6-channel final representation. And I'll go over what exactly those 6 channels are and how that's connected to instant segmentation. We can also output the resolution of our image. This LR just means that our model output is going to be the same size as our model input. We can specify the number of convolutions to change the size of this resblocks head. And then we specify the loss function, which in this case is a custom loss function from CytoDL to do instant segmentation, and then the post-processing steps. So as the output from this head, we'll call this act thresh label class, which lets you apply an activation, do some thresholding, and possibly label connected components. In this case, we're just going to change the data type to a float 32 numpy array. and then we're going to save out our images from there. Finally, for the model, we have some optimization parameters. So here we're just going to use atom W as our optimizer. You can pass in the learning rate and the weight decay. And then we're going to use an exponential learning rate for our learning rate scheduler. So since we're training this model not on full FOVs, but just on little patches, we're eventually going to have to apply this on a full image. We want to make sure that we're able to do that. So how we do this is we pass a sliding window over a large FOV. So we can specify the ROI size of each of those sliding windows. And this is referencing back to that same patch shape that we used in our data configuration file. We can also pass in the overlap, whether we want a progress bar and some other keyword arguments. Okay, so that's our model configuration. Let's go into the trainer configuration. So we use PyTorch Lightning to manage our training loop. This allows us to execute arbitrary callbacks during the training loop, and it makes everything very easy to optimize and scale. So we use this Lightning trainer class to manage our PyTorch training loop. We can specify the number of epochs that we want to run this for, the accelerator that we want to run this on. So this makes it easy to change this from a CPU to a GPU, a TPU, whatever hardware you happen to have. PyTorch Lightning makes it really easy to train your model on whatever you've got. Okay, so these are kind of the sub-configuration files that we've specified. And now we want to turn this all into a single experiment. So the experiment config basically includes each of these sub-configs and lets you do some overrides. So the experiment configuration file is the ultimate source of truth for what actually gets run with your deep learning experiment. So from the experiment configuration file, you can override any of the options that you've set below. So for example, if you decide in your experiment that you don't want to use the CPU, you want to use a GPU instead, what you can do in the experiment is you can specify trainer.accelerator equals GPU essentially. All right, so that's enough about the configuration files for now. Let's actually start training some models and visualizing our instance segmentations. Right. So our instance segmentation method is inspired by flow-based methods like cellpose, omnipose, or scoots. And what this means is that we kind of transform instance segmentation into an object similarity task. So what that means is for each object in our instance segmentation, we want to have one representation of kind of the core of that object. And then we want to have from each pixel in the image, the object that it corresponds to. So how we do this is our first channel of our 6-channel model output is going to be a connectivity preserving erosion that forms the core of each object. So you can picture this like two spheres inside of each other. So we have a large sphere and a smaller sphere inside of it. That smaller sphere is like the object core. Next, we have a semantic segmentation, which we generate by thresholding the instance segmentation. And this is going to be just a binary mask of that outer sphere. And then we have 3 channels representing the Z, Y, and X vector fields that predict the direction from each point in the semantic segmentation to the closest point on the object core. So again, thinking back to 2 spheres inside of each other, for each point in the space between the larger sphere and the smaller sphere, we want a vector that points to the closest point on the surface of the inner sphere. And then finally, we have a boundary loss that we can optimize using just like a classic segmentation loss. All right, so let's run this next cell, and this will just look at one of our FOVs, perform that pre-processing that we specified in our data configuration file, and then visualize one cell's example from that data set. So I'll run the top cell. And then we're gonna, load one FOV and examine one example cell instance segmentation. Okay, cool. So unfortunately, this membrane image is super dark, so it's a little bit hard to see. Hopefully you got one that looks a little bit nicer. But if we look at each of the 6 channels that's generated here, we see this object core, which is parameterized with a signed distance function. So basically each point in this image is going to be the distance to the background class. And this is nice because it lets us do varying thresholds to detect this object core during the post-processing. Channel 2 is a semantic segmentation of each object. And these are just 2D slices of what are actually 3D objects. Then we have gradients in the Z, Y, and X direction. The Z gradient one is a little bit hard to interpret since we're just looking at 2D slices, but you can see in the Y gradient that the top of this object is bright, meaning that the vector is pointing down towards the interior of the object, and same with the X gradient, the left side is bright, meaning it's pointing to the right. And finally, we have this boundary segmentation as well. All right, so that's our instance segmentation format. Next up, let's actually train one of these instance segmentation models. Here we're just going to train for 5 epochs. In practice, you would want to train for a lot more epochs. But what we're going to do here is we're going to load some of our FOVs and then return them in this format that we talked about before, where we have this list of dictionaries. Each one has a raw and a segmentation key. And then the value for that is going to be a NumPy array that we'll perform all of our data augmentations on. And then our actual function that does the model training is going to be very similar to what we've seen before, where we're loading this configuration file. We're going to apply some overrides, and we'll get into that in a second. And then we're going to train this model by passing in the data set that we want to train it on. So let's run that cell and then talk about the overrides. So the overrides here in the API, you can specify in the format of a dictionary. So what this is going to do is it's going to override the number of epochs that we're going to use to 5. We can set the output directory of our model where it's going to save out the logs and the example images. And then we're going to pass these overrides into our train model function. So let's run this, and then we should be able to see how our model is training. First, it's going to load the data set. Okay, so the first thing that's going to be printed out is some information about which devices are being used. So here you can see that a GPU is available and we're using it. These are all good signs. We can also see that we're using 16-bit precision instead of 32-bit. This is something that's configurable from the config files or the overrides. And the next thing that's printed out is some information about the model architecture that we're using. So this is the dynamic UNet that we saw in the configuration file, and then you can just see the parameter count for each layer in this network. That's a lot of information, but it can be useful for debugging to make sure your model is the size. Okay, so we didn't get to see it training because that took too long, but you can see that it ran 4 epochs, 2 steps per epoch. This is a very small training run. But if we look at a graph of the loss, which is loaded from the output directory that we specified earlier, you can see that our loss is increasing or decreasing for a little bit and then increasing. This is not ideal, but that's just kind of what you get when you only train for 5 epochs on a few images. Okay, so we've trained this instance segmentation model, and now we want to apply it to create some single cell data sets. So how we're going to do this is we're going to apply our instance segmentation model. This is very similar to how we trained it, but instead of doing model.train, we're going to do model.predict and pass in the data set. Again, we're going to pass in some overrides on where the data should be saved out. And here we're also going to include a checkpoint path. So this checkpoint path is going to be to the file that we downloaded earlier in those first few cells. This is a pre-trained model. So it's not going to be the one that we just trained. But let's run this, and it should predict on a crop from an FOV and show us the post-processed instance segmentation results. Okay, so that prediction is pretty quick. Now it's going to do some post-processing on the network output to generate the final instance segmentation. Okay, cool. So here's our example membrane image on the left, and then our predicted instance segmentation on the right. You can see there may be a few places where it could do better, but overall, this is definitely good enough to give us our instance segmentation to create a single cell data set. So we can use these segmentations to extract single cell images from our FOVs. There's kind of a cleaning process that we want to do to create these single cell images. The first thing is that anything that's touching the border of our FOV has incomplete information, so we want to throw that out. So we're going to remove all the cells that are touching the border of our FOV. Then we're going to normalize their intensities, and we want to exclude cells that are too small, which is probably just noise from our segmentation model, or too large, which might be like a merge segmentation. So we want to remove segmentation errors. And then we also want to remove sources of variation that we know aren't biologically relevant for our perturbation detection with the drug data set. So one example of that could be alignment of the cells. So we don't really care if a cell is at like a 45-degree angle or a 50-degree angle. So how we're going to address that is we're going to take the longest axis of each segmentation and rotate them in the y-axis plane to align the major axis of the cell with the x-axis. And then we can measure the size of each of our cells after they've been aligned and find like the 95th percentile of that size and crop all of our cells to a fixed size as just kind of pre-processing for our next step, which is going to be training the contrastive model on these single cell images. So this next function or set of functions is going to do everything that I just talked about: remove border cells, do some intensity normalization, and then extract these single cells from our FOVs. And then at the bottom, we should get an example of what one of these single cells looks like, so we can visualize what we're actually going to be passing into our contrastive learning setup. Okay, so right now that progress bar that we're seeing is just extracting each of the cells from our FOV. And then we see a nice example of one of our single cell images. So we have a data set of something like 20,000 of these single cell images. We just downloaded a few of them to train a contrastive model on this notebook. Contrastive learning is how we're going to be doing this feature extraction and applications for perturbation detection. The general idea of contrastive learning is that you can use this idea of similarity and dissimilarity to train a model that learns some sort of interesting unsupervised features about your data. So in this case, we're going to use VICReg, which is a relatively recent contrastive model that's shown good performance while being pretty simple to understand and implement, and also has just a bunch of flexibility. So the goal with contrastive learning, or one common format, is we take our single cell images, which is I here, and then we sample two different random augmentation pipelines. So T is the set of augmentations, and little t is going to be our sample from this augmentation pipeline. So we have two different augmentation pipelines that we apply to this single image I, and we end up with two different augmentations of the same image, X and X prime. We're going to pass these both through the same encoder to generate features Y and Y prime. And then we have this projector head, which basically takes Y and Y prime, which are going to be like 256 dimensional. And this projector head increases them to be 1024 dimensional vectors. And the VICReg paper that you can link here explains why this is actually an important part to prevent representational collapse and all that. So we end up with these features Z and Z prime, which we're going to use for training to calculate our loss on. And there are three loss functions for this VICReg setup. The most important one is this invariance loss. So basically contrastive learning, we're going to say we have two different augmentations of this single image. Your representations here, Z and Z prime, should be the same because the semantic content of these two different augmentations is the same. So one way that a contrastive model could kind of get around this constraint and do a perfect job at this invariance is by predicting the same set of features no matter what its input is. So that's where this variance loss comes in. So it prevents this representational collapse by saying within a batch, our features should be diverse. So this says like basically don't predict the same thing for every single image in a batch. And finally, we have this covariance term, which enforces that our latent features should be independent. So this says everything that's off of the diagonal in our covariance matrix should be close to 0. So these three variance, invariance, and covariance losses form up the VICReg method. And this is just an image from the VICReg paper. The choice of augmentations is really important. There are augmentations that you can imagine that would destroy the semantic content and would make this invariance part really difficult to learn by your network. Here we use random flips, random rotations, intensity shifts, Gaussian smoothing, and some slight resizing on the two different copies of our same cell. And we find that this performs well for getting a model that's interesting. So I'm not going to go into too much detail on our data config here, but basically it's implementing this pipeline that I just described, where we take two copies of our image and sample augmentations from them to create two different augmented copies of the same image. Yeah, and you can look at each of these data transforms that we have here and see how that links back to what I've just described in the VICReg pipeline. So our model is also going to be slightly different than from our instance segmentation because we're doing a contrastive approach instead of the supervised approach. So again, we have this backbone network that takes in the two different augmentations of our input image. That's going to be our anchor images and positive images. And then we have our representations y and y prime here, our anchor representations and our positive representations. And then we have a task head, which is our projector network, to provide those z and z prime embeddings that you saw earlier. And this is where we actually calculate the contrastive loss. So here, the main thing that I want to highlight is we're using this contrastive model instead of the multitask M2M model that we used earlier. But a lot of the keyword arguments are very similar here. Okay, so let's train this contrastive model. Here, I'll actually show the model training. Okay, here you can see the epoch increasing as this model is training. That's nice. You can see the loss is changing. That's also nice. Okay, cool. So our model is trained. Again, we're not going to use this model that we just trained for like 20 seconds. We're going to use a model that we pre-trained earlier and apply it on this data set. So what this is going to give us is it's going to give us a CSV with features for each of our single cells. So that was just a little batch, but you can see that we end up with the CSV that has these 256 features, which is our Y presented from the encoder, and each row is a single cell. Okay, so next we're going to start looking at our drug data set. So we have all these different structures, we have all these different drugs. Let's just encode these with our contrastive model and look at a PCA of each of these features to see where there might be some interesting differences here. So on the top, we're going to have the drug treatment that we used. And then on each row is going to be a different structure of interest. So some of these we don't have data for, but I'll highlight the ones where we do and where there might be something interesting. So here we have paclitaxel as our drug and tubulin as our structure that's targeted. In green, we have the vehicle, and in orange, we have the paclitaxel representations in the PC space. So you can see that there might be a slight shift here between our vehicle features and our paclitaxel features. So we might want to revisit that one. For Sec61B, which targets the endoplasmic reticulum, we don't really see any strong perturbations between the vehicle features and the Berfelden or the drug features. So we're going to ignore this one for any follow-up analysis. Same with beta-actin. There's no clear shift between the vehicle features and the paclitaxel features. But with the Golgi, we see for Berfeldin and on the right, S-nitroblebistatin, we see pretty significant shifts between the vehicle features and the drug-treated features. We're going to zoom into those in a second. This tight junction protein 1 was used as kind of a negative control where we knew that none of these drugs should affect it. And we see that in this representation of the features in PC space. So we had 3 combinations of drugs and structures that we think might be interesting to do a closer analysis on. So what we're going to do here is we're going to take these first 2 principal components that we've established here, and we're going to do a linear discriminant analysis on that. So what that means is we're going to find the axis that best separates the vehicle and the drug-treated distributions, and we're going to do a walk across that axis. So from like the vehicle distribution to the drug-treated distribution. And as we walk across that axis, we have what that corresponds to in the feature space. And we're going to look at the data set that is the 4 closest examples in the data set to that coordinate in the feature space. So we can envision what the model is actually seeing as we see the differences between the drug-treated and the vehicle-treated cells. So that's what this next cell does. You can see we're doing a visualization of the linear discriminant analysis here. Okay, so the first one we're going to look at is paclitaxel in our tubulin. So this red arrow represents the direction of our linear discriminant analysis. So we're starting kind of in the green by the vehicle cells, and then we're walking towards the paclitaxel cells. And you can see from this plot that our LDA is doing a pretty good job of separating our vehicle cells and our paclitaxel treated cells. And then if we go down to the bottom, as we traverse this LDA line, we can get the 4 closest examples to each of those coordinates. And there's a pretty clear trend as we move from the vehicle treated on the left to the paclitaxel treated on the right. The most notable thing is that the intensity goes down a lot over the course of this LDA walk. But there are some minor differences also that we might notice. So it seems like the cells on the right are a lot smaller than these cells that we see on the left. And it also might be the case that the bundles of tubulin are brighter. They have more contrast on the right than they do on the left relative to the intensity of the rest of the cells. So the idea with this is not to definitively answer like the effect of these drugs. We just want to have a better understanding of what's going on in these images in an unsupervised way that we can follow up with more hypothesis testing or actual measurements. Like cell volume is something we can directly measure, and we can validate whatever we are observing through this latent walk later to actually get some concrete findings. The next structure that I want to highlight is Golgi and treated with Berfeldin. So again, we have this LDA projection starting with the vehicle and moving over to the Berfeldin treated cells. And this is the Golgi. So over here on the vehicle, we see this classic Golgi morphology where we have these nice blobs and puncta. And as we move over to the Berfeldin treated cells, we see this total dissolution of the Golgi apparatus where the intensity is pretty much spaced out completely across the entire cell. And this lines up nicely with what we know that Berfeldin does, which is cause the disassembly of the Golgi apparatus. So it's pretty cool that our model is able to recapitulate that in this feature space. And finally, we have Golgi and S-nitroblebistatin. So again, we're moving from our vehicle-treated cells on the left through to our Berfeldin-treated cells on the right. And as we complete this latent walk, we again have this nice blobby punctate morphology of the Golgi on the left, and then a very different type of dissolution of the Golgi apparatus on the right, where we have kind of these really bright clusters with a very, very distinct morphology from our vehicle-treated cells. And again, this is something that wasn't actually identified in the original paper, but is an interesting finding that deserves more validation. So that kind of concludes our demonstration of CytoDL. The last thing I want to do is just highlight some other tools that CytoDL offers. So as I mentioned, we provide default projects for semantic and instance segmentation, classification, conditional GANs, label-free prediction, but we also have a bunch of unsupervised learning tools for contrastive learning like you saw today, masked image modeling like hierarchical and vanilla autoencoders, masked autoencoders with division transformers. We also have joint embedding predictive architectures, also called JEPAs, as well as some variational autoencoders on images, point clouds, and tabular data, and also some deep geometric models with rotational equivariance. And finally, we know that this is kind of a tool more designed for the experienced bioimage analysts and deep learning engineers. So we're also including and hoping to release soon a Napari plugin for human-in-the-loop dataset annotation and model training and validation, starting with our semantic segmentation models. Okay, that's all that I have today. I want to thank all of you for joining me here. The Q&A is open if anyone has questions. You're also more than welcome to join other sessions. I know this wasn't quite all of the time that we had budgeted for this. So yeah, I just want to thank you for coming by.